So my name is Dr. Chaya Murali. I'm a pediatric geneticist and postdoctoral research fellow. I see patients over at Texas Children's Hospital, and I do my research at Baylor College of Medicine. And my research is generally interested in the psychosocial aspects of genetic disease and how we take patient reports of their own experience and incorporate that into clinical trials and into different studies.
What is epigenetics and how is it defined?
So epigenetics has become a big buzzword lately, and a lot of people are interested in it and wanting to understand what it means. So epigenetics, when you think about kind of the etymology or where the word comes from, epi comes from the Greek, I believe, the word for upon, so on top of genetics. And so what epigenetics refers to is how we control how genes are used, or the word that we use in medicine is expressed, which genes we turn on and use at certain times of our lives or in response to certain environmental stimuli or things like that, and which genes are turned off. And that's what epigenetics means. And there's all these different molecular or really small microscopic ways that epigenetics happens. There's people studying all kinds of aspects of epigenetics, including those molecular ways, including when things are turned on or off and so on. And that's what epigenetics means.
So how is epigenetics different from genetics?
That's a good question, and the answer is a little bit confusing because epigenetics, in terms of generally thinking of the study of genetics, it's part of genetics, but it's separate in some ways as well. So if you think of genes kind of like recipes, right? They're instructions for creating different parts of our bodies. Epigenetics is not, so genetics is like the spelling of the recipe. Are the ingredients right? Is it telling you to do the right things? Epigenetics is whether or not you're using that recipe at this time of day or at this time in your life and so on. So at different times in our lives, we have to use different recipes to make different parts of our bodies, to make different hormones and so on. And so epigenetics is basically whether or not you're using the gene right now, whereas genetics is just understanding the gene itself.
What are some examples of epigenetics that we might be able to observe around us?
This is a great question. So one example is like within our human body, right? We're made up of cells, which most people kind of are familiar with. And all of our cells contain our DNA, our genetic material. Every single cell has the exact same genetic material, from our brain cells to the cells that make our hair, to the cells that make our skin, to our nerves and to our muscles and bones and everything. All of those cells, as you can imagine, are pretty different, right? Because skin is different from bone, bone is different from brain and everything, but every single one of them contains the same genetic material. How is it that the same exact genetic material can make cells that are so different? The reason for that is epigenetics. Going back to that analogy about recipes, we all have the same recipes in every single one of our cells, but our cells know, okay, if I'm going to make a liver cell, I have to use only these recipes. If I'm going to make a bone cell, I have to make only these recipes. And so that's an example of epigenetics playing out physically in our daily lives. Another great example is when you look at actually cats. So going into the weeds a little bit, as people may know, boys have an X chromosome and a Y chromosome, and girls have two X chromosomes. They're both the same. Now, because humans have two different numbers of chromosomes, or X chromosomes, it turns out that you only need one X chromosome to be active and working for you to be healthy. And so for women, our bodies, every single one of our cells, it turns off or deactivates one of our X chromosomes through some of the aspects of epigenetics. And so it turns out that calico cats, which we may be familiar with, those ones that are kind of patterned white, black, and orange, those cats are always female. And the reason for that is because the color gene, the gene that tells them what color to make their coats, is on the X chromosome. And because different X chromosomes are active in different cells, that's why they have patches of different colors. And so that's another example of epigenetics playing out in our daily lives that you can look at and see.
Are all genetic diseases purely genetic, or do some have an epigenetic component?
That's a great question. Most genetic diseases that we think about and understand well right now are what we call single gene disorders. One gene is misspelled or not working correctly, and that causes an effect on that one gene, which has some other effects and causes a disorder. There are some genetic conditions that we know are epigenetic, where the misspelling or some problem with a particular gene actually impacts the way we use other genes. And some good examples of epigenetic disorders include Prader-Willi syndrome, Angelman syndrome, Fragile X syndrome, and Beckwith-Wiedemann syndrome. Now, those are just naming a few, and it's not going to be useful for you guys to memorize them, but just as examples of certain disorders that we know are epigenetic. Now, there's a good possibility that a lot of single gene disorders also have some epigenetic aspects. And that's kind of, based on the fact that people with the same single gene disorder may have different ways that their disorder manifests or affects them. And so that speaks to the fact that there's probably some epigenetic stuff going on that's affecting the way different people express the same genetic changes. That's still an area of active research, and it's not something that we really fully understand yet. So I wouldn't say it's ready for prime time, you might say.